Hello friends, welcome back to my channel. So in this video I'll be talking about the other two aspects of the uh, processes that is done by the renal system for maintaining the blood pH. In the previous video I have talked about the renal regulation of blood pH, the first two processes, that is the excretion of H plus ions or reabsorption of bicarbonate. Okay, and in this video I'll be talking about the other two aspects or other two processes that is done by the kidney for maintaining the blood pH. Okay, and those processes are, for the third process that is done or that is executed by the renal system for maintaining the blood pH is excretion of H plus ion in the form of titratable acid. Okay, right? The major titratable acid that is present in the urine is the sodium acid phosphate. Okay, here in this process, okay, what happens is, now this is the tubular cells. Okay, it is tubular cells. Okay, this is tubular lumen and this is the blood stream. Okay, so inside the tubular cells there is a formation of the carbonic acid, which is formed from water and carbon dioxide. Okay, and the enzyme catalyzing this reaction is carbonic anhydrase. This carbonic acid then dissociates into H plus ions and bicarbonate ions. Okay, and in the tubular lumen there is presence of the disodium hydrogen phosphate. Okay, this H plus ions that is formed in the tubular cells that is released into the tubular, secreted into the tubular lumen in exchange for this sodium ions. Okay, this hydrogen ion that is present in the tubular lumen then combines with sodium hydrogen phosphate to yield sodium dihydrogen phosphate. Okay, so this disodium hydrogen phosphate and sodium dihydrogen phosphate, they are the basic and the acidic phosphate respectively, and this H plus ions, it is excreted in the form of acid phosphate, that is sodium dihydrogen phosphate. Okay, and this basic phosphate and acid phosphate, they are the urinary buffers. Okay, and the bicarbonate that is produced into the tubular cells is then reabsorbed back into the blood stream along with the sodium ions. So in this process of renal regulation of blood pH, there is a net regeneration of this bicarbonate ions. There is a net regeneration of the bicarbonate ions. Okay, and one more thing, this process occurs at the distal convoluted tubules. Okay, the secretion of H plus ions, it starts from the proximal tubules and continues till the distal convoluted tubules. Okay, now the last part or the next process that happens is the excretion of H plus ions in the form of this ammonium ions. Okay, now again this is the tubular cells. Okay, it is the blood stream and this is the tubular lumen. Okay, inside the tubular cells there is a formation of this ammonia. Okay, the ammonia is formed from the glutamine by the enzyme glutaminase. Okay, so during this reaction, the ammonia along with glutamic acid, it is formed, which is catalyzed by the enzyme glutaminase. Okay, the activity of this glutaminase enzyme is increased during the acidosis. Okay, so this ammonia, it diffuses into the tubular lumen. Okay. Now again, inside the tubular cells, the H2O and carbon dioxide, it combines to form carbonic acid catalyzed by the enzyme carbonic anhydrase. This carbonic acid is then dissociated into H plus and bicarbonate ions. Okay, the H plus ion, it is secreted into the tubular lumen against the sodium. Okay, in exchange for the sodium. So this bicarbonate along with sodium, it is reabsorbed into the bloodstream, and this H plus ion combined with ammonia to yield ammonium ions, which is excreted in the urine. Okay, so here there is also a net acid excretion in the form of ammonium ions, and there is a net regeneration of the bicarbonate. Okay, the activity of this enzyme, the activity of glutaminase, is increased during the acidosis. Thank you. If you like my video, please like and subscribe the channel.